Greetings, everybody. Welcome to the Evidence and Reasons for the Christian Faith video channel. This is a after show and a pre-show. We will be the primary topic, at least at the start, and we know how some of these shows go, is Dr. McLain's talk on mammal-like reptiles, and then some reflections on Dr. Tour's abiogenesis series so far. So if some of you would like to join the StreamYard panel discussion, I would be extremely grateful. So let me highlight something that I caught. Now, I actually know very, very little of paleontology. When I heard Dr. McLain using all these terms, I was totally lost. So that's just not my specialty. And it's something that I haven't really invested a lot of time in. People who know me know where my focus has been. So I wanted to say, I'm really glad to hear him talk, Dr. McLain talk about the nested hierarchy. There have been some creationists and even ID proponents. Oh, Nephilim Free, welcome. There have been some creationists and ID proponents who've insisted there's not a nested hierarchy. And I'm just like, okay, it may not be a perfect one, but if we take isolated proteins, definitely at the molecular level, I see it whenever I do gene browsing, can't run away from it. I've built them computationally. It's been part of my professional work to examine genes and proteins across species. And that, whether the nested hierarchy indicates evolution or not is beside the question. We can build them. Now, I will point out, and it's something evolutionists don't like to really talk about, there are conflicts in the nested hierarchies you build, because you could take one protein and you build a nested hierarchy one way, and you could take another one and you build it another way, and they don't quite agree. And there are other problems that the nested hierarchy doesn't solve. I've often said the solution to all this is that if the Earth, if the fossil record is young, if the age of life in general is young, there's no common ancestry. We've had independent origins. So Nephilim Free, welcome. Thank you for joining.
Hey, good to see you, Sal.
Thank
you do a great job of providing scientific refutation to the myth of evolutionism.
I'm sorry, I was my volume was turned down a little bit. I missed what you said.
Oh, I said you do a great job of providing scientific refutation to the myth of evolutionism.
Oh, thank you, and thank you for your work too, and thank you for joining this. I'd like to tell people that I have refrained from live streams over the last two or 3 weeks. Refrained from hosting them. I've participated, and the primary reason has been my mother's physical condition, but it's been improving. Now, one reason I'm really glad people will jump in on these live streams is in case I get pulled away to attend to my mother, the show will go on. Now, one thing we have going for us is this is going to be a relatively short show, because I know everyone here is eager to hear Dr. Tour give another episode of talking about the fraud in abiogenesis. Now, he doesn't use that word, but after last night's show, I was just like, oh my goodness, I didn't realize the fraud, the self-delusion. It's self-delusion at best. And he actually called one paper, what it said was outright lies. And just like, good on him.
Wow.
Good on him for calling a spade, because there's just such garbage in the abiogenesis community. They let stuff go that shouldn't pass peer review, and a valid criticism that should be recognized is suppressed. So now there are some things that are coming down the pike is very complicated as to what the purpose of the nested hierarchy is. And I'm going to first show, see if I can get it. I had some, Nephilim, while I'm grabbing this, if you had some thoughts, please.
Oh, well, you provided a link to join, and I just thought, oh, Sal's a great guy. I'll pop in. I didn't know if you really wanted anybody in the live stream or not, but you put the link out there to join. So I thought, well, I guess he does. So, I would point out that there are lots of discrepancies in the phylogenetics. The evolutionists don't like talking about them, but there are many incongruencies. And you know how difficult protein evolution is. So this idea that human and chimpanzee relatedness because protein codes allegedly show that, what a bonkers idea that is, right? Because you know as well as anybody that protein evolution is a mountain evolutionists simply cannot climb. But there are others, genetic similarities that don't match the anatomy or the morphology. The evolutionists will have admitted that the taxonomy, their taxonomy, the genetics doesn't seem to line up with their taxonomy, and they don't like talking about that problem.
I wanted, this is a little bit off. It's off topic, but then on topic in a way. What is your? Are you a heliocentrist, or do you have any opinion?
I'm a heliocentrist. However, I haven't dismissed geocentrism. I understand that geocentrism is illogical if you believe that gravity of the sun is what holds the planets in their orbits around it. And that would seem to match observable science. However, I don't discount God's ability to make it the solar system geocentric, and it appear to contradict everything we understand about gravity. So I'd say because God is capable of doing anything, it's possible that the solar system could be geocentric, but at present I'm heliocentric, I think, because of gravity.
Thank you for, I know that seemed like such an off-the-wall question. And Andrew, welcome. And I'll explain why I put it. I've been describing evolutionism as the modern day geocentrism. So when Erica gets on Modern Day Debate and say evolution makes predictions, I'm just like, yeah, geocentrism made predictions too. It doesn't mean that it's right. Yeah. So just because a model makes predictions doesn't mean that it's true. If a model makes predictions and some of those predictions come true, doesn't make the model true. If one doesn't account for all the things that fail, then the model is really questionable. So all this stuff with the nested hierarchy, I said, okay, it may explain the trees along, like, say, a single gene or a protein, but then it doesn't explain orphan genes. And this is, I really need to do a show on this. I asked Dr. Dan, hang on, can I mute someone? Someone's
hang on, I'll mute.
Okay. I asked Dr. Dan point blank, I said, do you believe all proteins descended from a single protein gene ancestor, gene/protein? He said no. And other people were weighing in saying no. And I'm just like, okay, so that's like an orchard model. You don't believe in common ancestry then for a gene coming from all genes. Now, they all believe, up until recently, they all believed in the single universal common ancestor of cells. But if some people had been listening to the kind of our journal readings, me and Emery Moyna, we've been finding that evolutionists have gotten kind of like non-insistent on that. They say, oh, maybe there could have been a pool of organisms that's our real common ancestor. It's like, okay, so no single universal common ancestry. It's just like, okay, So you're chopping the protein origin. It's following an orchard model. And now you're sort of admitting kind of a little bit of an orchard model. We're seeing it with prokaryotes, eukaryotes, and then even with the eukaryotic family, multiple origins. I said, just keep climbing up there, and it's going to look more and more like the creationist model. And so what evolutionists have said, well, why would God create nested hierarchies if he wanted to teach creationism? I said, well, this is like asking, I'm going to show a picture here, see if I could find it. I gave this presentation. There's someone trying to join the stream. I recognize the icon. Hello. Hey, Baptist Chad.
What's up? How are you guys
Hey.
doing?
Good chat.
Doing fine. So I'm going to show a, see if I could find, I made this presentation, and Stephen Jay Gould asked this question. Let's see if I could show it here. And I'm going to share my screen. Stephen Jay Gould asked, so there are two views. We could say creationism, and one way that it's been framed has been called the biotic message. And Walter Riemann was the co-founder of baraminology. He sort of had a falling out with some of the other baraminologists, but a lot of the people that were among the pioneers in baraminology recognized Walter's contribution. They gave him equal credit with Kurt Wise for founding the entire discipline. I did have an entire show on the biotic message, and his book is a real headache to read because it reads like a graduate level textbook. It's very thick and dense, and that's probably why it's not been well circulated. It is not for popular audiences, although I think he tried to make it for popular audiences. The way that he states his creationist theory is life is the product of a single designer. Life was intentionally designed to resist all other interpretations of origin. That is, life was designed to resist evolutionary and naturalistic origins. In contrast, the evolutionists like Stephen Gould, he'll say, did he meaning God, did he create to mimic evolution and test our faith thereby? And what Gould is referring to are things like the nested hierarchy, because if we superficially look at the nested hierarchy, it's sort of easy to think, oh, well, we sort of look like a related family, because you have, just like you have in family groups, you could see people look more related than others, and you have sisters and then cousins, second cousins, etc. And just by extrapolation, it's easy to conceive of biology coming from a universal common ancestor, but that is very superficial thinking, because we could look at a pencil, and when we dip it in water, it looks bent. Now, is God being deceptive by making the pencil look bent? You could see we could extrapolate the same thing to this whole question of nested hierarchy. Is God being deceptive because he made a nested hierarchy? Here, it'd be easy for someone to say, well, God's being deceptive by making the pencil look bent. It's like, no, he's not. No, he's not being deceptive, because if we look at it more closely, we can understand why this optical illusion is generated. And we'll realize the pencil isn't bent. This was actually a really difficult problem that probably lasted thousands of years until Snell, pictured here, codified the solution. Now, he wasn't perhaps the originator of the solution, but he certainly was a pioneer of it, and he codified it well, and he's the one who got the credit. So they call it Snell's law. Now, the reason I brought up geocentrism, and I felt kind of bad. I did this. Iger Television is, he's a very diehard geocentrist. And one time he just kept chat bombing me. I'm just like, I don't have time for this. We could have a conversation if you would have studied some of the physics. And I even put out things there that he didn't. He just totally ignored. I said, we can't have a conversation because it's really important to. This is a very good example of something that can look one way and it turns out being another. So yes, we can have a coordinate system where the Earth is the center of the universe, but there's just let me explain briefly the physics of it. You can feel when you're spinning if you just stand up and spin yourself and do that for a few minutes and then you stop, you're going to get really dizzy. You can tell if you're spinning. And there are all sorts of measurements that can be done in space where you can determine if you're spinning. There are a couple of effects. One, at the equator, the gravitational acceleration is a little bit less, but that's because of the spinning of the Earth. So if you take the same mass at the equator and then take that same mass and put it at the North Pole, they'll weigh differently. So weight is a function of the mass and the gravitational acceleration. Mass is an intrinsic property. That's one example. The other thing is the Coriolis effect. When we fire, people found out when you fire artillery shells, depending on what part of the Earth you're in, you have to make an adjustment for the rotation, the spinning of the Earth. So the Coriolis effect is also well established, and you have to account for it. The other thing is we could go into stuff like centripetal acceleration, and there's just more physics to this. So yes, we can create coordinate systems where the Earth is at the center of the universe, and we actually do that quite without even realizing it. Like we say, the sun rises and sets, and that's perfectly fine. But if we were actually trying to determine things in terms of like measuring accelerations and rotations, you can actually build instruments that can detect whether you are rotating in space, whether you're stationary. It has, you've probably heard the term centrifugal force or centripetal acceleration. That tells you if you're spinning. The Earth is generating centripetal accelerations, and you have to account for that. And we can measure kind of the kind of like it's related to the centrifugal force, except it's trying to force things outward as you're trying to spin it. So that's the best way that I could explain it. So why did I point this out? Well, the old geocentric model would make predictions that the sun would rise and set every day, and same with the stars. And so when I hear these evolutionists trying to brag about all the predictions they make and how they're fulfilled, I just laugh. I said, well, the geocentrists did the same. It didn't make their theory true. The reason that it eventually got discarded was we had all these anomalous measurements. So if they try to say, well, we have this nested hierarchy here, this is a proof. I said, well, what about all the anomalies? And I'm going to just list them for you. And the biggest one, and let me just try to illustrate this. Let's see if I could show this here. Promiscuous domains. See if I had one here. Let's see if I could show this here. This is the public portion of the paper I'm working on. Let me just give the audience a little bit of a tutorial on protein structure, and please interrupt me if you have questions. This is the human zinc finger protein, and it has 540 amino acids. This is the spelling of amino acids. So M is methionine. I think I should know what D is, and I'm embarrassed. S is serine, V is valine, A is alanine, F is phenylalanine, et cetera, et cetera. Now, this is it's very provident. This one has a very recognizable structure. You see it? Now that I colorized it, this protein has a very recognizable structure. This is non-random right there. These C's are cysteines, the H are histidines. They have to be pretty much, do you see the spacing that has to be there? And that's really critical to the way the protein functions. A lot of proteins of this variety are what they call transcription factors. It means it has to go into the cell and find an individual location on the DNA. Now I'm going to show you something with a nested hierarchy. By the way, there are other patterns here. If we use this Venn diagram, we can also group other things. This is a very non-random protein. It's provident we have one like this. So you could see this pattern here for the classical zinc finger. Now let me see if I could show. This is the collagen protein. 20 to 30% of your body, the human body's protein mass is collagen. So this is very important. You may see beauty products that advertise they have collagen to restore your beauty. I'm not vouching for that. I'm just pointing it out. And this has a non-random pattern. Do you see it? Should be very obvious. That's very important. This pattern is also extremely important to how it functions. It gives it very special structural properties. It's essential to what they call the collagen helix. And there's some other aspects of this. These are, we have a lot of synonymous codons. The G here that repeats glycine, it's actually implemented by different DNA codons, which is amazing. And let's just skip this. By the way, this shows the complexity. These are all the parts of the collagen protein. In addition to that pattern, there are little postal codes here. This thing will actually split off into 3 separate polypeptides / proteins at some point, and they become actually functional entities to themselves. Especially this one, this NC1 domain. Where am I headed with this? Oh, this is my other protein that I study, the topoisomerase protein. This one doesn't have an obvious pattern to it like the other ones, but it's also very complex. I should point out, just to give you an idea of the complexity, I'm going to highlight here, this DQ. Yeah, DQ stands for Dairy Queen, right? This DQ, that pattern was the subject of a PhD dissertation. This was so hard to decode what it actually did. So I'm just walking you to see how proteins, they're very different and all their various parts. This is a postal code here in the green and then another postal code in the red. Because this tells it tells the cell where to put this protein to localize it. So we call those localization signals. There are probably other terms for it. This is another diagram of what we call post-translational modifications. This was from a paper I did, poster session I did with Dr. Joe DeWeese, who's a biochemist, professor of biochemistry. So back to the nested hierarchy. We could take the human zinc finger and then take a zinc finger from a pig and then other creatures, and we can build this nested hierarchy. And I do have a point with all this at some point. And you're like, hey, that looks like universal common ancestry, right? So far, so good for the evolutionists, but it's going to break down here at one point. So you see, they have a very superficial view. They say, oh, that's obviously universal common ancestry. I'm just like, well, okay. And then you could do the same with the collagen. You could take collagen from a human and collagen from a zebrafish. Oh, by the way, funny story. I was giving a presentation and Dr. Sanford was in the audience. He said, what's Captain Kirk doing there? Captain Kirk is a human. I just thought he was just a nice representative of humans. So he did write me. He said, "What's Captain Kirk doing in your slideshow?" And so here's Humphrey Bogart and Ingrid Bergman. So for some of those who are familiar with old movies, that was also one of my favorites. So you could take collagen from a human and then zebrafish and other creatures, and you could also build a nested hierarchy. And the evolutionists at this point are saying, "Oh, this is so powerful. This is so powerful evidence for evolution." But then I'll confront them with this. I said, okay, I can get that this can change a little bit. You could see that the pattern here in the human is very similar to the pattern here in the zebrafish. If you actually examine this, you'll see that they're, you can even visually see how similar the proteins are without a computer, but then the computer will kind of lay it out like this. But then I say, okay, Mr. Evolutionist, what's the common ancestor of the zinc finger protein and the collagen protein? Can you evolve this from a common? Can you evolve a collagen from a zinc finger, or a zinc finger from a collagen, or can you describe the common ancestor? And I knew at that point, it's like, yeah, I got him at this point. They can't. At this point, they're going to fold. And I said, why don't they talk about this? So when we talk about microevolution and macroevolution, you can even hypothetically say, I'm not going to say that this is the case. You can sort of say, well, this looks like microevolution within the same kind of a protein. I don't believe it for other reasons, but just for the sake of argument, let's say that, the protein in the human and the protein in the pig, you can sort of imagine it came from a common ancestor. Common ancestor is zinc finger, but and you could do that for the collagen, but what you can't say is that this one and this one descend from the same ancestral protein. The sequences are just way too different. And then so I began asking around, and I asked even Dr. Dan recently. I said, so do you think all proteins, genes descended from a single common ancestor? He said, no. And this all began also in the summer. I asked Emo. He said, no. He said, that's stupid. So you reject common ancestry of that. And I don't remember what he said, but he effectively said yes. Then Speed of Sound weighed in and said, yeah, that's stupid. No common ancestry for that. And then Immutable Destiny. And then just one after the other, they're all saying no universal common ancestry for proteins. I said, that's great. What you've just described then is an orchard model for, let's see if I have a picture of this. I don't. I'm sorry. Apologies. You've just described my presentation blew up. I said, you've just described an orchard model for proteins. And you could see why this was a small victory, at least for me personally. It's like, okay, you're denying universal common ancestry here. Once a crack opens, that fissure can break and the flood can go through. Mm. And so that was, that's how I respond a little bit to this whole thing of nested hierarchy. I said, it's there at a superficial level, just like geocentrism. It looks like your predictions are holding up. But when you start to scrutinize it, it's like, oh my goodness, you can't have macroevolution from one protein family, major protein family, to another. You can have it within, you can hypothetically have it within maybe the same protein family with a lot of qualifications, but you're not going to have macroevolution across major forms. You can even see it statistically. And now they're openly admitting it. So that's how I began to deal with the issue. So why would God create this? First off, it doesn't show that evolutionism is true. It just means you have to look a little deeper and think about it, just like the old geocentric models. God wasn't being deceptive in making it look like the sun rises and sets every day, and therefore you might think that the sun orbits the earth. You just have to get a little bit more data and you'll see things more clearly. And that's what's happening in the present day.
Let me ask you a question, Sal. When it comes to, because you're more knowledgeable about this than probably anyone I know personally, and most creationists for sure. So would you agree that, or do you think that I'm wrong in this idea that when there is great similarity between the protein, let's say a protein in a human being, and a protein in a fish or some other or a mammal, for example, but there is also a difference. Let's say there's a difference of. Let's just take an imaginary example because I don't have one, an actual one. Let's
Mhm.
say that there's a protein in a human being that is, let's say it's 3,000 amino acids in length. Okay. So let's say there's also this protein in fish that's approximately 3,000 amino acids. And the similarity between the two is extremely high linearly for the vast majority of those two molecules. But yet there's a difference of 2 to 3, maybe 5%, or maybe even more, where they don't line. So the evolutionists automatically, because of their patternicity, they assume, oh, common ancestry, evolution preserved that information, right? But would I be wrong in saying that the great similarity between those two proteins exists because that similarity, the information that is similar, relates to a biochemical function, a need of the organism to process something in their environment, like other proteins, or to manipulate, to process potassium or B vitamins or anything like this. And that difference between them would relate more to the biological difference, the biochemical difference between the two animals. So the great similarity could be, there could be this great similarity in the two proteins between these two species, and that would relate to the class of protein that it is and the biochemical function that it performs. But the difference between the two would be related to the biochemical difference between the two organisms. What would you say to that?
The, um, it it is reasonable. It's there are we have I have some evidence, at least the proteins that I've studied. So when you talk to someone who says he studies proteins, usually he means either he's just looking at a whole bunch of them really superficially, or he studies one or two, and
Mhm.
they're both very important. Um,
Right,
the
because we
ones that
share, I mean, the classes, there's all kinds of classes of proteins, right? And they perform similar function in one
now
organism as another, that's what
now
I'm
I
pointing
have
to.
actually an example of that for you, and again, I'm the guy that is studying only one protein, at least professionally, for at least in terms of the papers that God willing, I'll have the privilege of being a co-author on that publishes in secular peer review. So let me show a screen here, and perhaps it will explain. And it also has some really interesting features I'd like to share with you and the public here, because I'm trying to, evolutionists keep insisting we're not special. We're just radiated form. We're not a special creature. This is biochemical evidence. We are very, very special. And I'll explain it. So these are the various, let me try to explain this diagram. We have this protein called called topoisomerase 2A. Top 2A means topoisomerase 2 alpha. And you could see that we're very, very, and then the next row here is the chimpanzee. So let me see if I could just zoom in here just a little bit more. Just, I apologize for the font, but can you sort of see it? I hope it's at least marginally clear enough. You could see
Yes,
row
it's
one is the
clear enough
human,
to read.
two is the chimp. This is a multiple sequence alignment. So you could see that we are really close. The coloring is kind of helpful. You could see how similar. You could see a stretch here where we're just like, as far as I could tell, we're 100% similar to the chimp. And I'll try to get to your question as we look through this. And I showed this protein earlier. Let me see if I could see it I showed the protein earlier. Did I have it? I had one. So when I spelled the topoisomerase protein earlier, like this is the entire topoisomerase protein. And let me see, this is, now we're just looking at sections of it, and you can see how similar we are to the chimp. But if you look, I don't have a fish here. Do I have a fish?
How many, if I may, how many amino acids do you know approximately there are in this particular protein?
About 1,500. In fact, I can give you the figure.
Okay, no, that's fine. I just wanted a rough estimate. That's good. Thanks.
Yeah, 1,500. About 1,500.
Okay.
About 1,500. Now, there is a region here where we're different than the chimps, but where it's really important here is, like, you could see the difference between us and the fruit flies. So, and definitely that in the yeast. You could see, like here, in this position, we have a T, which is a 309, and in the yeast, a P. And then there are other parts. I'm going to try to answer your question, but I'm just giving a setup here. There are these huge sections where there are differences, and this is important because what they did is there are actually two versions of this topoisomerase in humans, top 2A and top 2B, and you could take these, and they've actually done these experiments. It may sound a little gross. They took human genes and put them in a yeast and see if it would kill the yeast. So they took a human gene, put it in the topoisomerase, and put it in the yeast. The yeast looked fine. Then they took the topoisomerase B from humans, put it in the yeast, and it looked fine. And it's like, well, that's kind of funny. So why do humans have two versions of it that are slightly different? So even within a human being, we actually have two versions of supposedly the same protein. And what they determined, they did this experiment on mice because it's kind of unethical to be doing this with humans because you could kill them if you're re-engineering their genome. We do that with mice. The mice have a top 2A and a top 2B, and they're different. So what they did is they knocked out the top 2B and said, okay, well, if it's non-essential, the thing will live. Well, it died. And then they did the same with the mice top 2A. And it's like, okay, so the difference, even in the same organism, has significance to their biological function. Now, it's a little bit of a tough inference, but what you cannot do is take a yeast and put it in a human and expect it to live.
So
you can do the reverse.
You can
do the reverse. It's only one way.
Can I ask you something about that? Now, you said, was this a gene knockout experiment, you say?
Gene knockout experiment in the mice.
In the mice. And they knocked out the topoisomerase gene? Is that what they did?
They knocked out, we have two topoisomerase 2 genes. They're like 4 or 5 topoisomerases. Mm-hmm. I mean, that's how how they ended up numbering them. And they realized we have actually two versions of the topoisomerase 2.
Yes, but in the organisms that they knocked out, the gene, there's more than one in them also, a duplicate, a backup copy, right?
And this is very important because the evolutionists keep saying, oh, gene duplication, that's not a problem. It's like, no, you can't explain this by gene duplication because both are essential. And I can actually argue this for tubulin. If you don't have all the copies of these supposed duplicated genes simultaneously, the creature dies.
Okay, so that's what I wanted to get at. They also have more than one topoisomerase gene, and the knockout experiment eliminated one of them, and the organism didn't continue to thrive based on the other one. They went ahead and perished. Is that right?
Right. Now, the funny thing is, this is where I'm saying that humans are very, very special and mammals, is why then can we just take a human top 2A and put it in the yeast and it lives? Or just take the human top 2B and put it in yeast and it lives? See, yeast doesn't have multiple the proper term is paralog.
Uh-huh.
The
Can
yeast
I ask
doesn't
one
have.
more question?
Yeah,
Jeff?
yes, please. This is really
So-
good.
So they performed a knockout by eliminating one of the topoisomerase genes, and the organism died. Did they then perform a similar experiment knocking out the other topoisomerase gene and see if the organism likewise died?
Yes, yes. And oh,
And
by
it
the
did
way,
again.
a little bit of self-promotion. Dr. Deweese and I published this in a peer-reviewed creationist article, um, CRSQ. I don't know when it'll become public, but when it does, I can talk about it. So but I give a lot of credit to him. He's really the pioneer of a lot of this.
So they verified that both topoisomerase genes are necessary in the mouse also, or it
Yes,
died without either one. Is that correct?
right, right. And
Okay.
so I'm
All right, great.
extrapolating
Thanks.
it to humans because it's unethical to do this to a human. I mean, you don't just take a
Sure.
human, make a human embryo with knocked out genes as to do. That's why we ought to be grateful we have mice. And that actually leads to another point of the nested hierarchy. Evolutionists will say, well, why did God make all these creatures similar? I said, you ought to thank God every day God made creatures similar to you, because what would be the alternative to do medical research? We'd have to be volunteering humans and maybe even fetuses to do it. We have these other creatures we call model organisms. I said, doesn't that kind of turn things on its head a little bit more if you're just willing to maybe think out of the box? That maybe God's priority isn't totally to make it easy for us to see the creationist model. Maybe he wants us to understand that we're fearfully and wonderfully made. And we can do that by studying other creatures so that we don't have to sacrifice ourselves. So these poor innocent creatures can die in our place, just like they did in the Old Testament. And so you see this theme again of innocent creatures dying so that we can live and understand ourselves and our relationship to the universe. And just like, if the evolutionists would just be willing to think a little bit out of the box, they could totally see this. Now, the thing about the yeast, just to complete it, the yeast don't have two copies. They don't have a 2A and a 2B. You could actually see it here. It says top 2. That's because they don't have two copies. And that's the trick that they were doing. This is actually pretty difficult to do. And Emery's joining us. And we only have half an hour before James Tour comes on board. Welcome, Emery. So there's only, so that's why we could do this trick with yeast. We could put the top 2A and make it a substitute for top 2 in yeast, and the yeast thrive. Or we could take the human top 2B and put it in the yeast and it thrives. But it's obvious we can't do the reverse. And that's what I was saying. They keep saying we're just a radiation, the evolutionists will, and that, we're not special. I'm just like, no, we're way more, if you look at it from an evolutionary standpoint, we're way more evolved in terms of complexity, even along individual proteins. You could see this. This is an example. And one of the things that Nephilim free, if I could show you the difference, you could see the spelling here of the top 2B versus the top 2A. The top 2A doesn't even have that sequence. What that enables is it enables us to have these things called post-translational modifications. They're actually chemical modifications to the amino acid. They could put a phosphate group on the amino acid, and there are all sorts of other things like acetylation, and this is a methylation. So we're actually adding atoms and molecules to these amino acids, and that actually changes the fold and properties slightly. And so I've been showing this off because I said, when you look at it this way, it looks like, oh my goodness, every protein is almost like an individual memory chip. I'm just like, yeah, these are these little arrows here indicate positions that can be read, write, and erased. They're like, in fact, that's the term they use: readers, writers, erasers. And so now these positions here are very important. Apparently, the yeast doesn't care what all happens here. They
So
don't
that's
need it.
interesting, Sal, that the yeast survived.
Yeah.
But the mouse didn't. Can you elaborate on that just a little bit to help explain to us what's the true significance of that?
So the importance of this experiment says, okay, just because you could transplant one protein to another organism and it survives doesn't mean that the differences aren't important. because you have to do the reverse to really confirm it. Because the difference may not be important to the yeast. The yeast doesn't care. You could have a wide variety of variation in the yeast protein, and it's going to tolerate it.
And did I understand also that you say that they put topoisomerase, human topoisomerase, into the bacteria, and it was able to utilize that and survive?
Into a yeast.
using
A
it?
yeast is a eukaryote.
The yeast.
It's right here. It's this one,
Yes.
right?
So was the yeast cell able to use human topoisomerase
Yes.
to perform the same functions that it performs in its own self?
Yes. That was the big
Amazing.
deal.
That's bizarre.
Well, you see, that's half of the story. Everyone is saying, "Oh, well, then all those differences aren't really important." That was the first half of the story. We had a lot of evolutionists saying, "See, we don't really need all those positions." But then when we did the experiments on the mice with the top 2B and top 2A, and we did individual knockouts, it's like, no, that was a hasty conclusion that was wrong, because it's very important to a mouse. It may not be important to a yeast, but that does tell us one other thing. We are, if we accept kind of the evolutionary model, we're way more evolved. Mammals are way more evolved than a yeast. And that doesn't, any leaning to say that we're special really gets under their skin. And this is chemical evidence of it. I'm presenting chemical evidence, experimental evidence that we are special. That what I was trying to point out, humans are much more sensitive to mutation than yeast are. Same with bacteria. We're sensitive to losing genes. We're sensitive to mutations to individual. We're more sensitive to individual changes in our DNA than other creatures.
Well, we have
and that means we're very
systems.
special. We're complex. We're very complex.
Yes, well, we have intracellular systems, whereas the bacteria doesn't, or the single-celled organism doesn't. And maybe, what do you think, that additional tremendous complexity could be the factor involved here? What do you think?
Oh, absolutely. They have what they call the G-value paradox. If you look at the number of genes in a bacteria or a yeast, and then you look at it for something, a human, it's like, why do we have about the same number of genes? Not, I wouldn't say about, but like, we're not huge orders of magnitude more than a bacteria, which is kind of shocking. But then it's like, well, what do you mean? We have all these paralogs, but then it's the way the individual proteins are processed. We have alternative splicing. And then we have all these post-translational modifications, which kind of makes the protein almost be a slightly different protein each time we do a modification. And there are like 190 of these in the human topoisomerase. And that means you can have 2 to the 190th power different modulations of the same protein. And I got Dr. Sanford's forwarded me an email because he knows that my part of my work is studying post-translational modifications. It was a paper on the complexity of eukaryotes because they have post-translational modifications. They use it in very special ways. So when evolutionists talk about random mutation and that these are neutral sites, or you can dispose of them, I said you better be careful which experiments you cite. Because if you're just playing with bacteria and yeast, you could probably knock out a lot of positions. It's not going to compromise it. You do that to a mammal, it could be lethal. We are very much more sensitive to changes, which actually is a problem for evolution. Because if we're very sensitive to perturbation like that, we're very sensitive then to genetic deterioration, and natural selection would not select for the increase of complexity.
And think about, Sal, how that flies, and that post-translational modification of proteins flies in the face of what was known about genetics and the claims of evolutionists back in the 1950s and 60s by people like Niles Eldredge, with their simplistic view of organic life the only real information in the cell was the linear code for proteins, and that's pretty much it. Everything else is junk, and you know how they downplay the complexity of life. And here we've got this understanding now of post-translational information and function that showing that there's tremendous complexity in organisms beyond just the information that codes for the protein's amino acid string. I mean, wow, you know.
Did you see my? I think Emery and I had a journal club reading on cortical inheritance.
We did.
Yes, I argued there's good evidence that DNA is, and James Tour alluded to this in his abiogenesis talk. He was saying the RNAs are information bearing, the lipids are information bearing, the carbohydrates are information bearing. I'm just like, yes, he's up to date. He's actually up to date. When he was saying that, and he actually, I don't know, I would catch it because I'm interested in this. He said DNA has nothing compared to the carbohydrate code. The carbohydrates have way more information storage capacity. I've been saying yes. A lot of the information in the cell is in the cytoplasm, is in the carbohydrates, and it's distributed. DNA is only a fraction of it. And I also tried to say, okay, we take this 3.3 gigabases in the human genome. That would, if you do some calculations, it equates to about 780 megabytes. Do you think 780 megabytes, which is, my cell phone has more than that? Does that, just from an intuitive level, do people really think you could code something as complex as the human brain or the human immune system, much less an entire human being, with 780 megabytes?
Yeah, that's really ridiculous idea, and it ignores the compression and compressed nature of genetic information relating to proteins, how they're overlapping and nested, and that multiplies the information content of the genes that code for proteins too.
So I was just saying, and then on top of that, the evolutionists say, oh, maybe 10% is functional. And just like, okay, you think 78 megabytes? Do you think 78 megabytes is enough to make a human being?
You can't go to get a good exe file on a computer with 78 megabytes nowadays, just about.
Yeah. So now I wanted to say there's stuff that's there's stuff that is just on the horizon, and it's already in peer review. Not the labs I work with, but with other labs. They're like 40 labs and principal investigators quietly doing this. And this is just kind of a segue to, again, asking the nested hierarchy. Do you know how we actually do a lot of medical research? We have both ethical and we also have practical problems. If we study to understand human biology, do we just study human cells? There are practical and ethical problems in doing that. I mean, if you want to study embryology, it's better to do it on a mouse, because you shouldn't be doing that on a human being.
Well, let me ask you.
Yeah.
I'm sorry to interrupt. Go ahead.
So what I was saying, I was saying, God, why did you make all these other creatures similar to us? The creationist case would be so much easier if you just didn't do that. Okay. Yeah. I'm sorry that I'm just expressing, sometimes I get frustrated with the way, I may not always like the way God does things. Of course, I'm the one that's wrong, not him. Okay, I just admit that. But, all of us have these days. It's like, Lord, why? And that was how I was feeling. It's like, Lord, why did you make chimps? They look so similar to us and all these other creatures.
Yeah, I understand that frustration. I've felt it myself.
Yeah. But then I went to the NIH. I had to do a research presentation. Not my research. It was a student presentation. We did research papers, just like a journal club. But we would present to the class. We would explain the research paper to the class. We were given a postdoctoral fellow to work with us to help us make our presentation. And I was doing something on one particular neural receptor, and I asked, what are these cell types? He said, oh yeah, these are embryonic cell types. And I looked it up. This is from an aborted fetus. I just about wept. I said, so, and then I looked it up on the internet. They're selling these aborted fetus parts for $500 on the net so we could do neural research. I said, Lord, this just seems so immoral. And I felt so sorry for that human being that was sacrificed.
Creating a market out of that.
And I was just crying.
Yeah.
And then, it dawned on me. I said, you know what? We would have lost more, not that we had to lose any, if we didn't have these other model creatures like chimpanzees and mice. We can sacrifice them so that we could find healing. And that's a biblical theme. I don't think it's accidental that this is how we learn about ourselves, that these innocent creatures die so we could understand our human biology. I said, that's it. What we see as a progression is a stairway to understanding ourselves and other things in biology. I have some pictures of some intricate stairways where you can, I'll show it sometime. I'll give a separate talk on that. So what evolutionists describe as a progression, they call it a physical progression. It's actually a conceptual progression, and a lot of it's optimized for our benefit. So I'll give you an example. My professor here, Igor Rogozin, he wrote a paper. He's one of Eugene Koonen's staff workers. He said, paradoxically, humans share many more introns with the plant Arabidopsis thaliana than with the fly or nematode, which are animals, by the way. Mm-hmm. So there are, again, we have these odd things that don't quite fit the nested hierarchy, and that's one example. So we can actually study things about humans by studying plants. We actually learn things about humans by studying yeast. It's very nice to work with yeast because they replicate like crazy, and you could do all sorts of experiments. We actually began to learn about a lot of the huge research that helped the ENCODE project was studying yeast because it helped us understand chromatin. We had understand the human nervous system by studying things like squids and roundworms. And I was just like, wow, isn't this really cool? God gave little pieces of us and put copies of them in these other creatures, and we can learn about ourselves. And I said, that's how biology, that's the purpose of that's one of the purposes of the common design. I said, now that makes more sense. I was alluding to stuff that's on the horizon. We can actually now do a little bit of folding prediction in protein behavior by taking, I showed earlier, like a multiple sequence alignment of the topoisomerase. It turns out we might be able to predict chemical characteristics of proteins and also their structure based on just taking the differences and similarities. And there are labs around the country that are doing this. And I said, okay, so without the nested hierarchy, we couldn't really pull this nice trick. There's no reason nature would be so optimized to be giving us, kind of this, like a user's manual, if you just have a decoding machine, you could actually unwind it and you could predict protein structure, which is actually really cool because it's difficult to see protein structure. We have to do things like X-ray crystallography, but even then, that may not capture some of the really important.
Yeah, that's limited detail. Yeah.
So I'm saying, okay, so you want to ask why God made all these patterns of similarity and differences? I say, you look at that. Why is it optimized to help us? And you could almost, I'm just making just kind of a gentleman's bet. If you remove some of these creatures, hypothetically, you won't be able to make this protein prediction. It's like they almost have to be there so that we could do this. And that's kind of spooky.
Perhaps that's foreknowledge that God put into his creation to allow us in this era when he knew that we would have the technology to do such analyses that would lead us to provide for us a way of seeing that he's a majestic creator. Yes, I can see that, Sal. Hey, Sal, what time is Dr. Tour coming in? Let me know so that I can bump out before.
Yes, 17 minutes. Emery, did you have something to say? And
no, I literally just got back home and I saw you guys were live, so I figured I'd hop in. And you are talking about stuff that is very, very interesting, but I don't know a ton about. So, because I don't do a ton of work at this level. But
did everyone has their specialty? Dr. McLain was talking about.
Yes, I missed out on that, unfortunately, because I was at work out. But
well, for someone like me, that was just like I had no clue what he was talking about. The only thing that made sense was his when he had the nested hierarchy, and that inspired this whole meeting. I said I need to talk about this because he said, well, why would he alluded to the problem, and I credit him. He said, do we deny it or we hide? And then he gave scriptures, and I said, let me give my opinion from what I'm seeing, especially with what's happening in these labs. That nested hierarchy is a beautiful common design that is helping the scientific enterprise. It's like God is helping us along to understand ourselves and our biology. Mm-hmm. It's a pattern that shouldn't emerge randomly. And there are other things. So I don't know how many years away we are from making a convincing case, but there are laboratories working on this problem. For those that want references, look at direct coupling analysis. And I'll give a show on this at some point.
Well, they just keep producing more and more evidence that we were created, but they can't see the trees for the forest, That's the problem.
Oh, and good point. Really good point, because that was exactly what we were talking about. Let me see here if I could. Talking about they can't see the trees to the forest, which was exactly the point I was making, is that since you just joined, Emery, I was showing that even Dr. Dan is admitting now that there's no universal common ancestor for the proteins. Here's an example of why. So we can build individual trees for, like, say, the collagen, and we can build individual trees for, see if I could find it. Maybe I messed up the.
Well, while you're looking for that, I'm going to pop out because Dr. Tour is coming momentarily.
Yep.
And
all right.
Thanks
we'll
for letting me come in, Sal. We'll talk to you later. Keep up the good work, brother.
And thanks for joining us. So you could see a tree here for the zinc finger, and you could see a tree here for the collagen, but they can't proceed from the same tree. It has to be an orchard. Mm-hmm. And right. Even now, the other people, even Dr. Dan, Speed of Sound, or No Answers in Atheism and Immutable Destiny, emo, you name it, they're agreeing with me. There's no universal common ancestor gene, and you could actually kind of, I did this so you could actually visually see why. Mm-hmm. It's pretty obvious.
Yeah. The thing is, like, if you did a couple of phylogenies for, like, different animals using different genes, which they do a lot, you'd have the same problem. Like, you'd have trees that didn't overlap. I could probably whip this up in probably like 20 minutes if I went in and just grabbed a common animal and downloaded various different gene sequences, aligned them, and then ran them. I could probably create phylogenies that don't even overlap in something like 20 to 30 minutes. It wouldn't take me very long. It's not hard. All you have to do is use different genes, and you get different trees. And this is within organisms that are related. That's all you have to do. And so you can make phylogenetics tell you anything you want.
Oh, yeah, we had that discussion. That was really. Oh man, that was really. It's just hard not to laugh at them. I'm sorry.
I mean, look, I get what they're trying to do, but I'm sorry, it doesn't work. It doesn't do what they need it to do.
Yes, and what I was doing is I was relating it to heliocentrism and geocentrism. Mm-hmm. I said, if you have a very superficial perspective, you can get the totally wrong model. Mm-hmm. And I hear these evolutionists say, look at all the predictions it makes. I said, well, what about all the predictions it fails at? Mm-hmm. Yep. And geocentrism made lots of great predictions, like the sun rising and setting every day. It's like, oh yeah, it's consistent with the model. Look at all the predictions. But where it began to fail was stuff with like retrograde motion. And then later on, when we had precise instruments, things like the Coriolis effect and centripetal acceleration and centrifugal force, et cetera, et cetera. And it's like, I wanted to say, even totally wrong models will make correct predictions every now and then. That's not the issue. It's what do you do with all these anomalies that are total deal breakers? So Baptist Chad had a question. Type it in if you can. We have 10 minutes. I wanted to say just briefly before we begin, before we hop on to Dr. Tour's talk, I thought I thought he did us a great service yesterday. He actually called a paper and called out a paper and said what they said was a lie, and it's like good on him, because that whole industry, it's becoming cottage industry. It is becoming quite fraudulent, and they're getting away with stuff that shouldn't be passing peer review, and they're deceiving people and misleading them, and they're perpetuating their own industry. It's not very honest. At best, it's self-delusional. At worst, it's just fraudulent. And he called them out on it. Oh my goodness.
And unfortunately, like there is a very strong motivation for people to publish because of the way academia works. You either publish or you don't get tenure. And so like people will publish garbage, and sometimes they even know it's garbage, but they'll still publish it because they need to add to their, they need to add to their resume, and they need to add to their peer review history so that they can get tenure, so they don't, lose their job or lose their contract.
And get grants. And some of this is perpetuated.
No, it doesn't, Baptist Chad, not in the least. Collins is lying through his teeth. There are some similar broken things in humans, but okay, I guess what depends on what it means by broken. He's probably talking about pseudogenes. I don't know for sure whether that's what he was talking about, but odds are he's talking about pseudogenes. Yeah, turns out a lot of pseudogenes have functions, so they're not broken. And so this idea of shared derived mistakes is pure garbage. It's pure utter garbage from them assuming that the genome is 98% junk or whatever they say it is.
Well, the other thing is when Adam sinned and God said that cursed is the ground for you, there could have been a recreation of some sorts, meaning it triggered a few genetic changes across all species. I wouldn't be surprised if we had
I think
commonly designed damage,
that's possible. I'm there because we know there was no death. We know there was no death before sin. So there would have had to been at least some genetic breakage that's common to everything, so that animals with nefesh life could die. So there would have had to have been some kind of breakage, but who knows what that means? Like it's very difficult to pin that down unless you can pin down if you restored certain genes that they would keep things alive.
Well, what I would say is the bottom line is if we establish that the fossil record's young and that humanity is young, those are the essential things. If we establish that life is young, common ancestry is out the window. And there are some things with population genetics I just haven't had time to pursue. There's not enough intracellular variation among creatures, because when I try to find out, like, say, a well-defined creature, like, say, a particular kind of plant that's supposedly a living fossil, and I said, okay, look at the intracellular differences. Intraspecies, intra, meaning you take, once one individual of the same species and you compare it, and there's not enough variation. It's like, oh yeah, so either the molecular clock was wrong, or the most recent common ancestor of all of this particular tree in existence, or like, say, the horseshoe crab or the shark, which is a living fossil, their most recent common ancestor was like, say, 1 million years or less ago. And I said, why don't we try this with bacteria or microbes? And no one's doing this. And I'm just like, it's staring you in the face. There's a problem here. You don't want to admit it. So I think we're getting up close to.
We're about 5 minutes out at this point.
Yep. I didn't expect that this show would last this long, but and I'd like to thank all the audience who's been praying for my mother. One reason I'm now able to host live streams a little bit more on the fly. I can't do ones that's scheduled, like, say, a few days in advance. It tends to be just spur of the moment because, if she happens to be feeling well at that time, I'll go ahead and start a live stream. These scheduled shows are going to be just too tough, like, say, a big scheduled debate. But if it is kind of impromptu, it'll work. So I'd like to, I was just explaining why I've been able to do a little bit more of these shows. And I'd like to thank God for that opportunity because my mom is doing better. And I want to thank everyone for their prayers and their well wishes. Emery, my fellow graduate student in biology, I'll give you, please give us the last word before we watch James Tour's show.
All right, so just as a quick closeout, definitely head over to Dr. James Tour's channel. Anybody who's watching, he will be up. He'll be up and running in about 5 minutes. Probably a good half-hour debunk of the video that was put out by what's his name, Professor Dave. So, professor in air quotes. So I'll be over there. I imagine Sal's going to be over there. So definitely over to that. And thanks everybody for coming out. And I'll probably bounce around some more stuff with Sal at some point so we can nerd out about some more biology. So thanks for coming, guys.